Hey everybody, it's Alexander Williamson here with The Secret History Living in Your Aquarium. Today we're going to be talking about tissue cultures, and if you don't know what tissue cultures are, I have a whole bunch of videos on that. If you want to know how they're made, where they come from, how they're different than regular plants, if they're aquatic plants, if they're submerged, immersed, are they in some third dimension that we don't know about, fourth dimension, fifth dimension, and you know, what does a healthy one look like versus an unhealthy one, and how you would normally plant them. All that stuff, I'll link some videos at the very end, and you can just search through my channel, and I've got a lot of questions answered in some videos. But today we're going to be talking about, can you rescue a tissue culture? So say you're at the store, as I am often at the pet store, and I come across something like this. And this looks pretty toast. I would not pay for this probably, but frequently I'll say, do you think I could have it? And they'll say, yeah, we were going to toss it anyways. Especially if you know the people at the fish store. Even the big brand ones, you know, you can get them. You might be used to seeing them in these containers too. If they're not in a container with a gel media that has the nutrient gel in the bottom, or some sort of I guess antibacterial and nutrients, then you're not. It's called growth media in the scientific world of things. But if you don't have that, then it's not truly it's probably not a tissue culture plant in the retail scene of things anyhow. So what you're looking for when you're looking for a healthy one, though, is you want a plant that looks, well, healthy. And you're looking for, even this one's getting there, but you're looking for as much root tissue formed as possible. Basically, you want as many healthy plant nodes as possible, and then you can split every single one because they're clones. They are literally clone tissue from other plants, just like when you graft a plant or something. They take cells from where a plant grows, and it's called undifferentiated tissue, and they take a few of those cells and they put them in special chemicals, which, if you want to know the whole process, you can check out the other videos, like I said, that I've done on this, including a few live streams where we've gone through the whole shebang of how you do it. But basically, you add certain chemicals, and they're a mixture of nutrients and growth hormones, and you get those first cells, a mother cell, to start creating this kind of, almost like a tumor. It's like an undifferentiated mass of stem cells from whichever plant it is you want. So say you have an incredible plant, and like we talked about it in the variegated plant video recently, say you have a chimera, which is two different sets of DNA living in one plant. Well, say you want those, and you get a sample of both pieces of the DNA of tissue from both sides, and then you put that in the same mixture, you're going to get that growing that way. Or if you have a variegated plant that has a virus that is in its DNA that it passes on, then you can take that and you can pass on these one-of-a-kind traits in orchids, in aquatic plants, in the cannabis world. I mean, there's a whole lot of in vegetables. There's a whole lot of applications for it. But we're going to be talking about aquarium plants today, and we're going to talk about what we can do to save these suckers when they start looking really ragged. I mean, this thing should be lush full. There shouldn't be liquid in it. Should be gelatinish. But this means it's just depleted of its nutrients. There is not hardly anything left. The plants are starting to yellow, and here we've got a situation where it actually grew so much, it grew to the top of its bag and came out, and the nutrients are all gone. I don't even feel, no, there's a little bit of gel right there. But here we've got a stem plant. So when you've got a stem plant versus a rhizome plant versus a true aquatic plant, now, most of you probably won't be seeing true aquatic plants. That would be like Aponagitans, Aponjatons, however you want to say it, like Madagascar lace. That is a true one. The Thai lily, that's another one, or Thai lotus, whatever you'd like to call it. But the Nymphea, the Taiwanese Nymphea, that one I've seen as a tissue culture. And then there's also some Apelias and things that they sell as a tissue culture, like Siswasertong, things like that. But usually you're going to end up with either something like a sword plant or that has a mother and daughter and chaining roots, same with like some hair grasses and things, or you're going to end up with a stem plant with a whole root cluster. And in that case, those we can trim at the top, and this tissue up here, now that it's growing, it's just fine. These little sprouts, if we can get them not to melt, they'll be just fine. And there's some special conditions I'm going to have to tell you about to do that. Same thing when you've got carpeting plants. This one, though, is all dried out and crunchy, basically, plus the gel is spent. So what are we going to do about that? We'll talk about that. We'll talk about each one a little bit. But in the case of this one here, where we've got the big, tall growth, and you can see the top still, the plant, what it does is if it's not growing, it's dying. And so plants always grow up and towards the light, or over and towards the light. And if they're not doing that, like I said, they're dying. And they need to take the energy that they've already used in the lower levels, the lower branches and things. They're going to steal that energy, that nutrients, the carbon, the nitrates, the iron, the trace amounts of gold and zinc, and you name it, everything that they need, they take, they steal it, they utilize it for the new growth, because this new growth also has those undifferentiated cells at each intersection. So in nature, those are the places that can vegetatively grow. And vegetative growth is different than reproductive or fruiting, flowering growth, because when you're doing fruiting or flowering growth, that's when something needs to be pollinated for it to continue growing, and then you get a seed, and then the seed sprouts. Well, what we're talking about is vegetative growth with these, and we're spreading them via vegetative growth. And that just means you're cutting a piece off, whether it's from the root or from the stem, or breaking a rhizome in half, or scoring it in half with a knife. That's what we're doing here. And it could be anything from Bucephalandra to beautiful stem plants. So let's take a look at what we're going to do to save these, and what other products I'd recommend that you probably have. Doesn't matter the brand, but just that they fulfill that role. And then hopefully you can transform them into something more similar to what we're seeing over here. A whole lush garden of plants. There's another one over there that's probably too lush, needs a trim. So let's jump in right now and talk about all of that. All right, everybody, we've got everything we need right here, other than really healthy tissue cultures, right? That's what we were missing. So first, you're going to need your drink of choice. I like Dr. Pepper, so that's for you. Put that in you. Delicious. Pay me, Dr. Pepper. All right, next we're going to need some distilled water that's room temperature. Now, the reason you want distilled water, or at least filtered water, drinking water, is because it's going to have a lot less bacteria than using aquarium water. And tap water has chlorine in it, which actually is pretty hard on tissue of any sort, be it your fish's gills or plants. So you're going to need some water. You're going to also need some scissors. Boom, boom, boom. You don't need crazy scissors like these. I just happen to have some aquascaping scissors. In fact, those are probably terrible scissors for this use right here. All right, so now we're going to pull out the plant. You can see what's going on here. So this is Alternanthera renicii is the Latin name. You might know it as scarlet temple plant, but it is slimy. The gel that is the nutrients, as well as antifungal, antibacterial, and anti everything, antimicrobial. Technically, you should wash your hands first, or hit them with a little bit of rubbing alcohol or ethyl alcohol, whatever you got handy that will sterilize your hands and not have a residue, so something that evaporates. But then we've got this mushy mat. These roots are toast. They are probably, there are some white ones that are sinewy in here still that may be okay, but they're probably going to melt. I mean, let's pull this apart. We're dunking this in the water. Now, the gel does come off if you've got healthy stem plant or healthy tissue cultures. The gel does come off a lot better in warm water. But when you got tissues this old, you're going to find that this happens. You get chunks that just fall off. All these leaves are useless. They're dead. They're worse than useless. If we take these stems and look at them individually, this stuff is just waiting to fall apart. basically connects water and a little bit of nutrients. But it's probably that gel has probably been out of nutrients now because it's probably been in this container for 2 months. So now what we're focusing on with any stem plant, so any plant that you're going to plant that has roots, not rhizomes or bulbs or like a rosette, like when you have a flame sword or a melon sword, something like that, or Logonodrome boldi or Barclay longifolia. We're going to pull it apart and find the useful pieces. This, you could say maybe this is enough. You're going to want to find at least an inch, if not 2, of tissue that is still firm, and we're going to actually cut it off at a node. So it would be right here, right where these leaves are, right below that. And this is one piece. We can kind of dip it, make sure that all the kind of tissue is off. This piece is right at the border. This may or may not make it, but it was free, so we should give it a shot. So we're going to set that aside. I like when I have big tissue cultures, I like to use a basket like this so that I can dry it like a salad spinner. But basically what you're doing in here is you're just washing off all the dead leaves and all the stuff that's already melting. Now the melt occurs, like I said, because these plants, they have to keep growing, they have to keep living, or they're going to die. So we want to separate this tissue from the other tissue. And even though we have some pink plant going on here, you can see that this is starting to melt at the base of these leaves. So you want to cut right above where the melt stops if you can find that spot. Otherwise, you could try it here. And what's going to happen is this is going to deteriorate into ammonia and carbon and whatever it's made out of, all the nutrients and minerals, and that can actually poison the tissue culture, the soil that's in where we're going to plant it. So we've got, this one has a pretty good shot, I think, because see, we've got some root runners starting here again, and we've also got some green tissue, because remember, green is the only color that is going to be able to photosynthesize. If we had all pink or all white or anything like that, if this was a Pinto Anubias, forget about it. They die really easy. So when you need to be careful, and if you are getting an opportunity to pick through some of these, sometimes they'll have a pile of them, or they'll still take one free or whatever. They'll have groups of them, and you'll want to look for ones that are not variegated, which means multicolored on each leaf, because those tend to be weaker. They tend to not have chlorophyll in those leaves that are white. You might have seen that in house plants, or maybe you've seen my other video on variegation. But with the stem plants, you can kind of tear a little bit, and you can, if you want, you can then later use tweezers and plant each one of these. Now these are going to be really clean, these ones here, but it's just they don't have any roots starting yet, so we'll need to encourage that. You can see this one here actually does have a root, but these are both putting out runners, now that I look at it, all the way up at the top. So you need to be careful that that runner, if you put it in water, that doesn't pull the whole thing down. And also you need to be careful that you don't have fish like Corydoras or Autosynclus, or just even fish that are supposed to eat algae. You don't want a ton of them in the tank, because they are going to be eating any of this tender stuff, and snails too. Snails, shrimp, they'll all eat this. Now, the other thing that's going to happen in your aquarium is if we do this all completely submerged or underwater, aquatic growth, this white tissue, if any of it's left on, so you're going to want to cut it all off, if any of the white, or let's open another package too so we can see what we're looking at. So here we have Prosa, what is this one? Proser Pinaca palustris. Yeah, mermaid weed, I think, right? And this one is just really fried. So this one here, I mean, you can pour out this nasty liquid. That is just, I mean, and this one is molding. So something either broke the seal, or finally one or two of the little microbes that were in there, the spores, started growing. And so with this one, we don't want to contaminate the whole plant any more than it already is. And we definitely want to get rid of the mold. So we're going to take any of the shoots that are green, and we can pull those off. Now that has no chance of growing probably because it's yellow. The way it would suck up water and thus nutrients is totally fried. So the only way these stem plants like this are going to gain any strength and grow is going to be through photosynthesis and through absorbing, not through their roots, but rather their roots are fried at the moment. So they're going to only grow through absorbing up in their capillary, or if you're British, capillary action. This, just throw this away. It's moldy. It's not going to hurt you, but it's no good. So then we rinse off the roots, and we're going to pull off, again, it can be black, it can be red, it can be clear. They use different color gels that are full of nutrients, and different nutrients have different color profiles, and sometimes it's done for their inventory sake, sometimes it's done because the actual nutrients is that color, and that's the way it is. Maybe it's rich in iron or something. But this already just loosely going over it, we've got all the gel off, which is important because it's taxed anyways, but we can see that the roots just fall away. And so we might as well just have a trimming from the top of a plant in an aquarium. That's all this is. So from that whole tissue cup, this is all we're going to get out of that. Where normally, if it were a healthy cup like this one here, you would have a whole plant and all the little nodes, wherever you see the little squigglies, or you see little, let's find another example of that, where you see like things kind of condensing in the root structure. It almost looks like spaghetti or top ramen or something pushed together. This is each a plant cluster that will grow, hopefully, into a successful plant. But when you're rescuing, this may be all you get out of a cup. But it was free, right? So who cares? Let's carry on. So say you've got all that, and you've dealt with, now I've shown you how to deal with the root plants. Let's take a look at something a little different. Something that grows with a mother plant and then kind of carries on from there. It like spreads, like a carpeting plant. So this one isn't as badly fried. Grasses also, you may know, that they can get pretty dry, and you may see this in your own yard in the summer if you have a lawn or something, but they can get pretty fried and they can still bounce back, and they store their nutrients a little differently, and they also have, instead of having forked off like leaf nodes, they store their nutrients where the root and the stem meet. That's where they're, not their nutrients, but they're undifferentiated cells. And so sometimes you'll see these yellow strands that are completely dead all the way up, and those are done. Those are done for, and they're just going to add complication to your tank. So if you see a big chunk of them, just, rip them off, pull them out. They'll just come out like that. And sometimes you can just wash them in the water like this. This will get the last bits of any of that gel or agar that's in there that's nutrient rich off there too. See, here's a big chunk that's yellow. We don't need that, so we're going to pull this little chunk out. And even the roots are brown or dead, yellow or fried. So we don't need that. And the root ball below that is going to be brown and toast too. So we don't need to worry about that either. We're going to get these knots of dead roots kind of out. And then from these are going to be the biggest challenge of all the things we look at today. I mean, we're not looking at a whole lot, but these, I like to just kind of unfold them, break them in the center, and then I'll have a row like this of the plant, and I'll plant the whole thing if it's not so healthy, because it uses redundancies in the root network. So there's multiple pathways that this plant has established between wherever it started, the node that it started on, and the rest of the plant. So there may be multiple nutrient centers, and they may break and they may reestablish. But I like to grow it in a big long line. If it's really healthy, then you've got roots that look like this, where they're white and healthy and they're firm, they feel springy. Then this you could cut into pieces and literally just set on top of the aqua soil, and it would be all right probably. But that's how you're going to handle the same with if you have a sword plant or something. You just peel away like an onion the layers of bad stuff until you get to the healthy stuff. and that's what you're going to use. With this one that we started looking at earlier, this one, I know it looks really, really bad, but we don't need the roots anyways. Even though these roots are complete mush, it doesn't have bacteria in it. But what it does have is it's going to have carbonic, nitric, and ammonia, or carbonic, nitric acid, and ammonia in it. And that's just from the plant decomposing in a sterile, it's not completely sterile, obviously, because sometimes it grows mold, but it's from it breaking down in a sterile condition. It's basically protoplasm and goo and water and the nutrients of life. So this may remind you, so see this color here? Well, check out this. I want you to look at their system at ADA, which is who makes that tissue culture. So for nutrients, you're definitely going to need some nutrients, liquid fertilizer, because none of those plants that are stem plants are root feeders. But what does this look like? It's kind of a brown, sludgy looking stuff. Well, this is their brighty green mineral. So basically, you're going to need all your macros, other than you don't need a bunch of liquid carbon. That can cause things to melt, but you don't want algae. So in theory, if you have a product that's getting rid of the algae and you know how to use it, you can keep doing that in the aquarium you're going to introduce these into. But what's more important, in my mind, with unhealthy ones like this, is to put them in a nursery first, then transfer them to your main aquarium. And you have two options when you do that. And you can either float them in your aquarium just like this with a strong light right up top. And then what you do is you dose the water column with these fertilizers, at least as directed, which is, a pump for every 5 gallons daily or whatever it says on your brand. I happen to like this. It's a little spendy at $20 a bottle, but this is what I use for my best results because there's no nitrates in it. And like we said, that liquid that's already getting made in this cup from stuff decomposing is going to have ammonia and nitrates and nitrites and all sorts of decomposing amino acids and things like that in there. And we don't need that because extra biological material is just going to give food for algae or for rot to occur. So we want to strip all that and just get living stuff if we can help it. And then if you're going to grow it by floating it on the water, it's just important to make sure that you don't get algae starting. Now, algae is not going to start if you have no nitrates and no phosphates. So basically, if you're using a once all-in-one type fertilizer, it's probably going to have nitrogen in it. It's probably going to have phosphate in it and things like that. And that can sometimes start all this nutrients that's sitting here. Oh, there's a good piece right there that we just got. It's going to start it growing into a problem, into algae. So let's not do that. Let's avoid that. Sound good? Good deal. So same with this one here. This is like another, this is a crypt, and this one still has a lot of gel in it. So with this one, this is I believe this is a Cryptocurrene Axelrod, I believe, looks like. Maybe it's a Wenti green or something, but it looks like Axelrod to me. This one is actually pretty healthy. I'm pretty impressed that I scored this one because this side was facing out. I'll just cut away that and then treat this as normal. So see my other videos for how I'd normally treat it. And then look at this one too. Same here. We've really just got, this is at the end of its shelf life, but it's not toast yet. It's still got really bright white and firm roots. And you can see that there's not that many melted leaves. There's a few where the nutrients can't make it up because the stalk is melted. So just as you wash it in water, you'll see that those pieces come off in clumps. All the unhealthy stuff will separate from the other stuff just kind of naturally, just as you kind of tease it with your hands like you're giving it a bath. And if we've got something like, for instance here, African water fern, Bolbitis, this has a rhizome. So let's take a look at what's going on here. Let's cut this bad boy open. Yeah, let's jump into the bowl of water first, but then we'll cut it open. All right, now we'll cut it open. So This should be showing off above, like high up, but it's not. So this plant is so used to, right now, its life of being a dry plant. So a tissue culture is neither aquatic nor on land. It's neither submerged or immersed growth. It's in between. It's this weird clone state that's in a hermetically sealed package. And once you introduce it, so it's got some silica gel or whatever it is to keep the moisture out, or silica rocks. Don't put that in your aquarium. But basically wash off whatever's on there. This right now, you can put this in a terrarium or in an aquarium. This rhizome growth here is going to, this is a Java fern, like I said, and you can kind of tease it apart, but all these were started by little, they take a pipette and they put it on a tray and they try to hit it with 5 or 6, sometimes 10 different starting points, and then they hope for a mother stem kind of like this to develop. But each of these in theory will grow into its own plant if it survives. It's just, it has better chances when it bundles the nutrients together like this, and the light is all in one place, and you can make sure that's doing okay, and then also that you can trim any dead stuff, and it will actually sometimes graft onto one another, and you'll see the rhizomes attached to one another, and that can lend a hand to struggling tissue. Whereas these ones here, they're going to be on their own. These ones that are so thin and anemic. Now this plant package probably got dried out. These rhizomes never fully formed in the package. Only one really did. They should all be at least this formed, if not better. And same with same with Anubias and other plants that have rhizomes. But with this, really, you can just keep this either in a humid condition and keep the bottom ideally in a fertilized substrate, or you can float it, or you can tie it to a rock or something. I mean, really, these are super easy. These, you just kind of hope and pray for, and they get their nutrients from the water. They don't get their nutrients in the root. So like we said before, if it's a rhizome plant or a bulb or whatever, you can't do much for it other than cut away any dead material and hope for the best and feed it in the water column. If you want to feed the water column, I think it's best to do to pick a specialized dosage regime like these are my choice, and to do these daily, a little bit daily rather than a big dose weekly. You're gonna have a lot more luck with that. So then what we're gonna do is, if we have an aquarium, I'm gonna try this one as a dry start. So what I will do, even though it says dry, we're gonna put cellophane over the top here, and we're going to flood the bottom with water. Now, this is ADA Amazonia, so it's full in nutrients. It's Amazonia 2, which means it doesn't have raw ammonia in it. It does have nitrates and nitrites, which if we had fish in this tank, we'd have to worry about. So, we're going to plant this, something like this. We're going to plant each of these stems in here. Each stem will plant. And we're going to give them, what you want to give them is as much space as possible. You want to keep the temperature as cool that's in their zone of survival. So if it's a tropical plant, that means keep it around 70 degrees, not 80 or 85. Keep it around 65, 70 degrees in your aquarium. You want good flow so that all the old and dying tissue can get washed away, and so that the nutrients can soak into every little crevice of the parts of the plant that still are receptive to getting nutrients. And, with these, we're going to take these little stems and we're going to have the tweezers, and we'll basically be putting them down every few inches, and they will be laying flat. Won't look pretty for a few weeks, but they should perk up, and those will then be immersed. If I were to fill it with water, they would be submerged. But I want to try this, because if I do this, that means then I can change way less water, and the nutrients can be way more concentrated I can just take, 10 squirts of a nutrient formula, and also I don't have to deal with the algae as much. But even better than all that is the light doesn't dissipate through the depth of the water, and algae won't be growing on the plants. Now, mold, fungi, same thing essentially, and bacteria, those can definitely grow. And that's why it's important to wash the excess nutrients off in the agar, because that is basically a petri dish for them to grow on. So that's why we washed our hands. That's why we're washing it in the water that's, getting it clean, getting rid of all the dead stuff, and getting rid of any of the fungi or the mold that's on our plants. If we do all of this correctly, we will end up with a beautiful tank like this one. Sorry about that. Hold on. Look. All right, so in this tank here, you can see how healthy plants, they don't have any of those melting spots. They do put out runners when they need nutrients here. You can see over here, you can even see a running plant too, where they'll put out little nutrient nubbins that need to go into the substrate. But they're fine, they're doing well. And what we're gonna grow basically is this. We're gonna grow these kind of plants, immersed. So it's just the difference is we just aren't gonna have this, we aren't gonna have that much water. We're gonna have this much water. And we're gonna have this much water, and we're gonna grow out our plants, and they will be immersed. So then they'll need to convert in this case. However, you can just put them straight in the water. The risk you run though is that they might melt like this. And if they do that, it's just really important to keep the ammonia at 0, the nitrates near 0, but they need food. So make sure they're getting that from the substrate and nowhere else. And that's basically the advice I have for you. You'll get plants that can grow much quicker too out of the water. And what happens is first they'll wilt, like these ones. See the end of this? It's wilting. But once they get into the air, it's like having CO2. So if you don't have injection CO2 for your tank, start immersed, which means growing the roots in the water in the substrate, but the plant above water. Because then it can just breathe the air, and it can get its CO2 from the air, which is in the air we breathe anyways, because lots of life breathes it. And you can also get the nutrients to the base if those roots are healthy enough for it to stand. If not, you're going to want to submerge some of the leaves so the leaves can still absorb that down low. But in the air, they're getting an oxygen exchange and the CO2 exchange, plus there's no water column to make things dimmer. I mean, down here you can barely see. It's very dim, very dark. Up here, everything's bright. So light, every 6 inches you go down in the tank, you lose 50% of your light. So pretty extreme decrease in these LED lights as you go deep into a tank. So a shallow tank with water this much flooded in and substrate below that is going to give you your best shot for growth. And then you're just going to need to make sure that there's either a power head in there or something moving water around, a pump, a little fountain, and that will move water around, and then you're going to want to clean out all of the debris as it builds up over time. That is how we're going to rescue these. So for a follow-up video to this one, I want you guys to check in. By the way, here's a healthy Java Fern rhizome. Really chunky, thick, looks like wood. It's kind of reddish orange. But I want you guys to check back in if you're interested. I'm going to do a video probably in a month or two of if we grew these out properly, how many survived and how much tissue culture we got from each. See these, what I'm doing right here is I'm floating these. You can also do this with those tissue cultures, but you need to make sure it's in a tank with no algae bloom. If you're prone to algae, you're going to get the algae and it's going to melt your plants anyways, and then they can't suck up nutrients. But by floating them like this, we can get those little sections, the nodes with undifferentiated cells that don't know what they're going to be yet, we can get them to put out root cells at any point in a stem plant. So any stem plant or any rhizome plant, you can float like this and just let it get its energy. So that's essentially what we're doing by immersing it, is we're floating it, but some of the leaves are going to be up like this. The rest are going to be laying against that substrate. The roots will grow down in and the plant will reach up, and then when the roots start growing out, they'll feed the rest of the plant, but until they do, this will be submerged in room temperature water. So I hope that makes sense, and I hope this helps you guys save some, maybe score some free plants. The other thing you could do too, like I've got a little pre-filter sponge here. You can also take some of these things and get a coarse pre-filter sponge and actually grow moss, as well as some of these small rotalas and things. You can grow them right off of there. The other trick is you take a heater, like one of these little heaters, and you put it in a bowl of water. You put it in a bowl of water within your aquarium and then put saran wrap over the top. That heater is going to heat up, not boil it or anything, but it will evaporate the water quicker than room temperature would and keep the humidity up in this tank that has cellophane on it and your water and your substrate. So that is my best advice, is to do, you can look it up online too, the dry start method with plants you're trying to save. And since we're not going to be putting any fish in there, go ahead and just bombard it. I mean, just nail it all with nutrients. Just give them all the nutrients they can handle, just not the ammonia. You'll burn tissue culture plants with ammonia, but give them all the other nutrients they need as long as it's not causing algae. So that is my word of advice for y'all. I hope you have a wonderful time trying to score some of these free plants, or maybe they're $1 a tissue cup or something, then they're totally worth it. And I hope you guys have good luck. Let me know what you guys do. If you have any other tips or tricks, I'd love to hear it. The Barr Report by Tom Barr, his website's a great place to look for info on this, as well as Dennerley and 123 Grow and Aquascape. There's so many great websites and producers of these tissue cultures now, and they all have different tweaks on what they say you should do, but hopefully one of those leads is a good one for you, and you'll be able to get the most out of your plants this way. Thanks for joining me, and if you liked it, hit that thumbs up. You sat around a long time with me, and if you didn't, well, I don't know why you're here, but thank you for joining me, and I will talk to you next time. If you want to see an update video and be alerted when that comes out, hit the little bell, obviously, and say you want to know alerts in the future, or some alerts in the future. Whatever it is you want from me, I am here for you. And we're coming with videos usually 4 times a week, as well as 2 live streams. So it's a busy channel with a lot of nerdy info, but that's the way we like it here. Talk to you guys later, next time on The Secret History Living in Your Aquarium. All right, talk to you guys later.